Hello, my name is Marianna E Horn, and I'm going to share research on hatching time and embryonic learning in fathead minnows. Predation risk is an important driver of ecology and evolution. Prey species evolve different defenses against predation. We often think of evolved morphological defenses, such as spines or a shell, but these defenses can also be induced, like the shift in body depth of this crucian carp. Another induced change can be in life history, such as the timing of hatching, where prey can hatch either earlier or later to reduce overlap with predators. Many species use behavioral responses, such as freezing or hiding, in addition to other defenses. Prey animals must choose between the risk of being active in the presence of predators and the reward of foraging or mating opportunities. Lost opportunities do come at a cost, and so an organism can benefit from being able to evaluate threat levels to avoid missing opportunities when the risk is very low. Some organisms use the intensity of injury-released alarm cues to estimate the degree of risk. Others may evaluate the relevance of these cues based on the ontogeny of injured conspecifics. This helps them to avoid responding to predators that are not relevant to all age groups, which may happen as prey outgrow predators and move through different prey guilds. Learning to recognize predators allows organisms to have flexible and up-to-date knowledge of the threats around them. Many animals, such as fish, can learn to recognize a novel predator in the presence of injury-released alarm cues. Conspecific alarm cues trigger an innate fright response. And if an individual identifies a novel predator odor at the same time, it will later be able to identify that new odor as belonging to a predator. My PhD research focused on whether or not this type of learning can occur in embryonic fish. I exposed embryonic minnows to different concentrations of injury-released alarm cues that were collected from adult minnows. I wanted to see whether they could learn in a threat-sensitive manner and whether this exposure had an impact on their hatching times. I bred pairs of adult minnows and collected 120 newly laid clutches of eggs for these experiments. Starting 24 hours after deposition, I exposed each clutch of eggs to one of 6 conditioning treatments. My 3 control treatments were distilled water, predator odor, or alarm cue. My 3 learning combinations each combined predator odor with a different concentration of alarm cue, either low, medium, or high. These alarm cue exposures simulate predation events and as such represent stress events. Other stressors, such as elevated temperature or low oxygen, are known to cause early hatching. And I was curious if this predation stress would have the same effect. In fact, Cush and Chivers 2004 shows that minnow eggs hatch early in the presence of crushed embryonic conspecifics. I wanted to see if the same early hatching would happen with our adult minnow cues. I checked the development of each clutch every 4 hours. To avoid pseudo-replication, I counted each clutch, rather than each egg, as a replicate. I noted when the eye spots developed, when the first eggs hatched, when at least 90% of the clutch had hatched, and when all viable eggs had hatched. This gave 6 measures, and because they were related, I used a principal component analysis to combine them into a single synthetic variable, which I used for analysis. After the fish hatched out, they were allowed to grow for a few months before I tested their responses to water or predator odor. I filmed their responses in a visually occluded tank and quantified movement in line crosses and shelter avoidance for 5 minutes before and 5 minutes after the injection of a stimulus, and calculated proportional change in behavioral metrics. The stimulus was either a water control or a predator odor. One would expect that fish that learned to recognize predator odor as a threat would later reduce movement or decrease shelter avoidance as fright response to that predator odor. Fish that received water rather than predator odor, as well as those that did not learn to fear predator odor, would not show fright behaviors. First, I'm going to share what happened when we tested the juveniles a few months after hatching. The embryos that were exposed to combinations of alarm cue with novel predator odor did learn to recognize it as a threat, and that learning was in fact threat sensitive. This means that the anti-predator responses of the newly hatched juveniles depend on the intensity of the alarm cue they experienced when they were learning as embryos. Learning conditioning at all 3 concentrations resulted in reduced movement in response to predator odor. Only those that learned at the highest threat level added a second, more drastic shelter-seeking behavior, demonstrating a notable behavioral shift in response to an elevated threat level. Now let's take a step back in time and look at the hatching data. As you can see, our adult alarm cues did not have an effect on the hatching rate. The last data set I showed with the learning demonstrated that these embryos have indeed identified this adult alarm cue as indicating a threat, or they wouldn't have learned from it. However, they are not hatching early, as minnows are known to do in the presence of embryonic alarm cues. This suggests ontogenetic specificity in the hatching response to alarm cues. In addition to showing that minnows hatched earlier in the presence of embryonic and dietary cues, Cush and Shivers actually showed that there was a physiological cost to hatching earlier, specifically a reduction in size at the time of hatching. If eggs are being eaten, it is worth being a bit smaller if hatching out earlier means avoiding that predator. However, if it is adults that are at risk, then staying in the egg is clearly the safer choice. Embryos hatching early in response to adult alarm cues would experience a mismatch, so this ontogenetic specificity is advantageous. My research addresses fundamental questions about embryonic exposure to predation threats. I have demonstrated that embryonic minnows are capable of sophisticated threat-sensitive learning and ontogenetically relevant responses to alarm cues, including not hatching early to cues that represent a threat to adults, while retaining that threat information for use with juveniles after it becomes relevant. I would like to thank the many people who contributed to this research effort and the University of Saskatchewan and NSERC for funding.